Welcome to all of you. We are continuing the lectures of carbohydrates, and today in this segment, probably segment third, we will have some discussion on the isomerism. Isomerism, we'll be discussing isomerism in carbohydrates, and second thing which we will discuss, what are the features and functions, biological significance. What is the biological significance of glucose? Biological significance of glucose. These two topics we will take in this video tutorial. The first thing is isomerism. Probably I have shown you one part of isomerism in my previous lecture. That was Enantiomerism. Here we will discuss what are the various types of isomerism which are possible in case of glucose. If we see the first thing which we have the first type of isomerism is these carbohydrates may show stereoisomerism. Stereoisomerism. One. Second, they may show enantiomerism. Enantiomerism. Then third, they also may show diastereomerism, and they may also show epimerism, and the last is they may show anomerism. These are the various isomerisms which are possible in carbohydrates, and we'll take them one by one. The first thing is stereoisomerism. Although they are quite related to each other, but with some differences. We will take first stereoisomerism. What is stereoisomerism? Stereoisomerism is that type of isomerism in which the isomers, the two isomers, 1 and 2 isomers, they differ. The isomers differ in the spatial arrangement of their groups. I mean to say, isomerism means they need to show something common. And here, in case of stereoisomerism, the molecular formula is same. The molecular formula is same. However, the groups, the adjoining groups, differ in the arrangement in the space. If we see, for example, we have something C here, it may have A here. We have another isomer C, it may have A somewhere here. So the arrangement around the space, arrangement of groups around the space is different. For example, I will quote one example. We have glyceraldehyde, and we have dihydroxyacetone, glyceraldehyde and dihydroxyacetone. Both the two have the same molecular formula. But when we have to make this glyceraldehyde, we see it is carbon 3, 1, 2, 3. It is here, functional group is CHO, here it is OH and H, and here it is CH2OH. This is your glyceraldehyde, right? And when I have to make dihydroxyacetone, again it is 3 carbon, 1, 2, 3. It has a ketonic group, here it is CH2OH and here it is CH2OH. Now see the spatial arrangement of the groups around this central carbon, or see the spatial arrangement of group anywhere. The spatial arrangement is different, so they are stereoisomers. Those isomers which have the same molecular formula but differ in the groups in the space are known as stereoisomers. Then the second thing which we have, that is the second part, we have enantiomers. What are enantiomers? Enantiomers are those isomers which are mirror images of each other. They are mirror images of each other. Same molecular formula and arrangement is such that they are mirror images. Mirror images means what is the requirement for enantiomerism? The requirement for enantiomerism is the presence of chiral carbon. That is the presence of chiral carbon. Probably I have discussed it earlier. For example, if we have a carbon, it is attached to E, it is attached to D, it is attached to F, it is attached to G. Then when the carbon is attached to 4 different groups, we call this as a chiral carbon. So here we have a chiral carbon because it is attached to 4 different groups. And such compounds which have a chiral carbon are basically showing enantiomerism. Now we have one more thing which we need to understand is that when we see the carbohydrate, there is chiral carbon. You just made, right now we have made this glyceraldehyde. For example, it is CHO, it is C, H and H, and here it is CH2OH. Just see whether this carbon is chiral or not. Whether this carbon is chiral or not. On one side it is attached to OH, right? On the other side it is attached to H, and then here it is CH2OH, and here it is CH2OH. Yes, this is a chiral carbon. This carbon is a chiral carbon. And this has the capability, if we have a chiral carbon anywhere, it has the capability to show the enantiomerism. And these enantiomers are mirror images of each other, and you know they have a capability to rotate the plane polarized light either to the right or to the left. Or when they rotate towards the right, we call them as dextrorotatory. When these compounds, when these isomers, the type of isomer which is turning the plane polarized light towards the left is known as levorotatory. So we have dextro and L, and I told you most of the sugars are dextrorotatory, right? This is one more thing. Then the second we have diastereomers. Third part is diastereomers. What are diastereomers? Diastereomers. Here we have third, that is diastereomers. Diastereomers. Again, these are those isomers. These are those stereoisomers. Enantiomers are also stereoisomers. Diastereomers are also stereoisomers. And diastereomers are those stereoisomers which are not enantiomers. Diastereomers are those stereoisomers which are not enantiomers. Enantiomers. It means those isomers which cannot rotate the plane polarized light to right or left, which are not mirror images of each other, are considered to be as diastereomers. So we can say diastereomers have a different physical properties. They have a different physical properties. They differ in their physical properties, and on the basis of which we can separate them. Physical separation is possible when they show the different physical properties. Accordingly, we can separate them physically. So this is diastereomers. So we can say all stereoisomers which are not enantiomers, we can say like this, all diastereomers which are not enantiomers show stereoisomers. Or we can say all stereoisomers which are not enantiomers must be diastereomers. Right? This is one more thing. And then the fourth is epimers. We have discussed epimerism. Epimers we have already discussed it. I told you epimers are those isomers which differ around the configuration of one hydroxyl group around a particular carbon atom. Yesterday I told you we have D-glucose, and we have D-mannose. D-glucose and D-mannose are what? Epimers, because they differ CHO 1, 2, 3, 4, 5 and 6. It is CH2OH. Here it is OH. Here it is OH. Here it is OH. And here it is OH. This is D-glucose. Beautiful. D-glucose. And I told you when I had to draw the mannose, I had to change at carbon number. I have to change the things at carbon number 2, 1 and 2. Here it is CHO. C, OH should be on this side. Then C, OH on this side. C, OH on this side. C, OH on this side. And CH2OH. Yesterday I had discussed this thing. I told you this is D-glucose, this is D-mannose. They differ in the configuration of OH group around one carbon atom only. See, just see this. Here OH is on this side and OH is on this side. So we said we call them as epimers, right? Then we have the last thing, what are anomers? What are anomers? What are anomers? Anomers. Anomers are the sugars. Anomers are the sugars which differ in the configuration of OH group. Which differ in the configuration of OH group around the anomeric carbon or around the top chiral carbon. What does this mean? I told you, let's see this anomers. Last one, anomers. Anomers are those isomers. They are those isomers which differ in the configuration of OH group. Which differ in the configuration of OH group. They differ in the configuration of OH group around. Epimers also differ in the configuration of OH group, but I have not mentioned at what carbon atom. It may be at it may be at carbon number 2, it may be at carbon number 3, 4, 5, anywhere. So far as epimers are concerned, they differ in the arrangement of OH groups, but they differ in the arrangement of OH groups around any carbon atom. But anomers are those isomers which differ in the configuration of OH group around anomeric carbon. Around anomeric carbon, or we call it as top chiral carbon. Top chiral carbon. I will see it this way. For example, I would like to make D-glucose. Right? This is D-glucose. Okay? This is D-glucose. And you know, this D-glucose has OH on this side, suppose. OH on this side. OH here. And here we have CH2OH. Alright? This is your D-glucose. D-glucose. Not so beautiful. I am poor in this drawing. But, okay, probably you can understand. D-glucose is basically having OH at the anomeric carbon, the OH is on the lower side. So the anomeric carbon is this carbon, which is closed functional group. This carbon, top chiral carbon or anomeric carbon, is the carbon number 1. This is the carbon, this is the anomeric carbon. We consider this as anomeric carbon because it is attached to the functional group. And here, when the OH is on the lower side and H will be on the upper side, very obvious. Here also there will be H, here also there will be H, here also there will be H. We have discussed this thing. And this is the alpha D-glucose. Alpha means the hydroxyl group which is located at the anomeric carbon is on the lower side. And if I will tell you to design alpha D-glucose instead of alpha D-glucose, let's make beta D-glucose. Where is the difference? The difference is only at one place. Suppose I will see how I can draw the beta D-glucose. When I have to draw the beta D-glucose, everything remains the same. Here, OH is here, and position 2, OH is 3 at in, and 4, it is here, and here this is 2OH. But the difference lies at the anomeric carbon. The hydroxyl group is on the upper side and the H group is on the lower side. So in this example, we have a difference in the spatial arrangement of hydroxyl group. There is one difference. This hydroxyl group is located on the lower side, and in the second isomer, the hydroxyl group is on the upper side. We can conclude that there is a difference of OH group around anomeric carbon only. This is the anomeric carbon, and OH group is differing at this carbon only. For the rest of the carbon, it is everywhere same. So we can say this is anomerism. This way we can conclude what is isomerism, what are the various types of isomers which are possible in case of carbohydrates. Then we will move to another part. I told you we will discuss another part. That is the biological significance of, or we can say biological significance of glucose. We will be taking now monosaccharides one by one, and we will do what is the biological significance of glucose. Okay. See, biological significance of glucose. This is our next topic. See, glucose has a structure. I have made it open chain structure, and if I will see the ring chain structure, it is in the pyranosic ring, stable in pyranosic ring. I have a structure like this. We have OH, we have OH, we have OH, we have CH2OH here, right? This is the glucose, and this glucose, everyone maybe illiterates are also known. They have also, glucose is known to illiterates as well. Everybody knows about glucose. First thing is, the glucose is also known as grape sugar, also known as dextrose, also known as blood sugar. So many names. And glucose is the most common, most widespread, and most important sugar. Right? Common, it is everywhere. You see in the blood, our predominant sugar is glucose. We have never discussed about fructose, never talked about any other sugar like mannose and galactose. So it is very common. Everywhere you see glucose. We start from the bacteria. And we go to the human being. In this wide range of organization, you will find glucose everywhere. And this glucose is the common, widespread, and important sugar, right? And one more thing, when we see this glucose is stored in our body in the form of glycogen. Although the glucose is basically produced from the plants by the process of photosynthesis, right? But it is utilized as a respiratory fuel by the animals. And this glucose may be burnt in the body of several organisms. It may be burnt aerobically, using the oxygen, utilizing molecular oxygen. It can be burnt. And it can be burnt anaerobically, sometimes aerobically. Sometimes we see in the muscles we utilize glucose. We burn the glucose anaerobically at some times. And it may be utilized by fermentation. We have seen there are a number of industrial processes like alcoholic fermentation. And we do fermentation of molasses, we do fermentation of sugars, and this predominant sugar is the glucose. So this fermentation is usually carried by the microorganisms, maybe in the gut, maybe in some experiment, in some experimental model, or in some industrial process. We are seeing glucose is being utilized for making so many things. Fermentation. So this is one thing, it may burn aerobically. Second thing is the glucose is basically converted into glycogen by the process glycogenesis. Glycogen means glycogen, genesis means synthesis. So glucose is converted into glycogen in our body at two places. One is liver and second is the muscles, right? This is stored, and in this stored form, for example, if we will take the fast, maybe voluntarily, or maybe the circumstances may be such that we are supposed to be at the fast, we don't die. Do you know our brain requires the important respiratory fuel, that is brain. Our brain is working continuously. We cannot afford to switch off the supplies to the brain for 1 minute even. We will die. So the efficient fuel, or you can say the most important fuel, by which the brain works, which is the utility, which is utilized by the brain, is the glucose. And this glucose passes the blood-brain barrier. It It is not fructose, it is not mannose, it is not galactose, it is glucose. And when we will go for the fasting, maybe it will be 72 hours fast, 2 days fast, I can say, we are not dying. Our brain is not stopping working because there is no neuronal symptom. Reason is that the stores of glycogen present in the muscles, present in the liver. We are utilizing that. Our brain is utilizing that. He's taking the glucose from that. After that, still we do not die. We do not have neural symptoms because we are not in a state of hypoglycemia. After that, what is happening? It is the ketone bodies. It is the ketone bodies which are utilizing. It is the fat which is being mobilized and is converted into the respiratory fuel. But if we continue the fast for much more time, obviously there will be nervous symptoms when there is a state of hypoglycemia, and this hypoglycemia will cause the nervous systems which may be sympathetic as well as parasympathetic. Starting from food seeking to some we can say to food seeking to some tumor, I can say, or ultimately it may lead to conversion, or ultimately it may also lead to we can say complete death. So the main fuel, I am here pointing out, the main fuel is the glucose. It is the glucose. It is the main respiratory fuel, and this glucose is utilized from the bacteria to the human being. Every organism is utilizing glucose. The question is, if glucose is a monosaccharide, we do have other monosaccharides. We have fructose, a monosaccharide, a hexose, we have also mannose, a monosaccharide, we have galactose, a monosaccharide. All these are monosaccharides. The question is, why the glucose is used as a preferred fuel for our body? The simple answer for this is the stability of the glucose structure. When we see the glucose has less tendency, it has a less tendency for glycation. What does this mean? Compared to the fructose, mannose and galactose, glucose has a less tendency for glycation. And now what is this glycation? Glycation is addition of glucose units to proteins or fats. I mean to say, when we add carbohydrate in the form of glucose to the protein, the process, this is a protein, we will add carbohydrate to the protein. And which type of carbohydrate? Glucose, for example. It will form glycoprotein, maybe. Glycoprotein. This process is known as glycation. There is another process, and a very important process, known as glycosylation. Glycosylation, very important process, very important. Glycosylation means addition of carbohydrate residues to proteins. Addition of carbohydrate residues to proteins to form glycoproteins. And here, this process is known as glycosylation. The same process I have represented just above. I am telling you, when there is addition of carbohydrates to the proteins, it will form glycoproteins. It is known as glycation. And I am telling something else here. When there is addition of carbohydrate to the protein and it forms glycoprotein, it is known as glycosylation. What is the difference between the two? In glycosylation, the glycosylation process is superspecific. The glycosylation process is superspecific and requires energy. We have a number of glycoproteins that are functioning in our cell, in our body. We have membrane, and on the outer side of the membrane we have glycoproteins and glycolipids. These glycoproteins and glycolipids are helping in recognition. They can discriminate between the self and non-self. So when we say there are number of glycoproteins in our body which are very, very important. Number of glycoproteins are there which are very, very important for our body. and we need to synthesize them. We need to synthesize them. Our body needs to synthesize them, and without their presence, our metabolic system is not going to regulate. We'll fail to regulate if we do not have glycoproteins in our body. But the question is, it means the addition of carbohydrate residues to the proteins in a specific manner, and where there is requirement of energy, where there is formation of glycoproteins, which are extremely important for our body, is known as glycosylation. Then what is this process? This process is there is addition of carbohydrate to the protein, but not specifically, but non-specifically. It is not a specific thing which body needs to do. It is something which the body does way of compulsion, by way of compulsion. Our body is taking the process of glycation. Our body is adopting the process of glycation at the moment when there is very additional amount of glucose, and we are not able to get rid of this extra glucose, and this glucose has a tendency to get bind non-specifically with the protein and forming glycation. By the process of glycation, it forms glycoproteins. And remember, we know when the glucose levels are high, glycation does not take place normally. But glycosylation is the normal process which is taking place in our body and is very, very important. Glycation is taking place when your blood glucose levels are very high. For example, I will tell you, we have the patients of diabetes, and the diabetes, the long-term symptoms, the long-term, we can say complications of diabetes are, we have neuropathy, we have nephropathy, we have cardiovascular diseases, we have also we can say this retinopathy. There are a number of pathies which are associated with this diabetes, and it is being presumed that these pathies, these complications are basically by the process of glycation. Our proteins are being misled, our proteins are being changed, and these proteins are being changed by the extra amount of the glucose which gets attached to our useful proteins and makes them useless and makes them functionless. Which are the extra amount of glucose are getting attached non-specifically with our proteins, with our lipids, and it will alter the function of that protein and lipid in such a way that our body will not perform the normal function which it was supposed to do. So in diabetes we have the problem, the glycation is the very hazardous problem which is taking place, and it has a large number of ill effects, right? So this is one more thing I told you, glucose. Yes, my question was, glucose has why the glucose is the main respiratory fuel, why not fructose, mannose and galactose? Yes, this was the question. If we see the tendency of fructose, mannose and galactose for the process of glycation, is many times more than glucose. There's a question, why glucose is so important? Why glucose is the main respiratory fuel? Why it is the more abundant? Because this glucose has very less tendency for this abnormal glycation. Whereas when we compare it with the fructose, mannose and galactose, they have a higher tendency to adopt the process of glycation. Therefore, if fructose would have been our respiratory fuel, then our glycation process would have been very rapid, and our damage to the body would have been very fast. So that's why glucose is an important fuel. It was Emil Fischer who gave the structure of glucose and received the Nobel Prize in the year 1902. Right? There is one more thing which we call as glycemic index. Glycemic index. Glycemic index is something. This is used in case of glycemic index term is used. I can tell you in diabetic patients, because this index tells us which food has more sugar and which food has less sugar. Glycemic index, I will tell you. Glycemic index it is a tool. It's a tool to measure how much amount of glucose is raised by a particular food. I will tell you. It tells us, for example, if we take some food, what will be the amount of the glucose? What will be the amount of the glucose which will increase in our body immediately after taking the food? This is index. Because I told you this is very important in case of diabetes. We cannot afford a high glucose level, and sometimes we have to see whether this food has a high glycemic index. It means once I will give this food, there will be a steep curve, there will be a steep rise. There will be a steep rise in the glucose in our body. Steep rise in the glucose in our body. And we have given glycemic index a value, arbitrary value, 100, a value of 100 to the glucose itself, to the sugar itself. We have given him a value of 100. And we are comparing other foods with the glucose. For example, if some food we have, it has a glycemic index of 14. That means it is glycemic index is 14. It means it will make a peak. Suppose this is glucose. It has a glycemic index of 100. The curve will be like this. It will raise the blood glucose level up to the 40% as compared to the glucose. And we have a food who has a glycemic index of 90. So we can say it can raise the blood glucose sugar level up to 90% as compared to the glucose. If glucose would have been fed, it will be 100, and if we give this food, it will be 90 only. So glycemic index, by this way we can understand. We have put the foods under different ranges: low glycemic index range, less than 50. We can say high glycemic index range in foods, more than 70. So on that way, that's one more thing which can tell you about glucose. So glucose is stored in our body, I told you, and glucose is the constituent of large number of important polysaccharides. It is the constituent of starch, it is the constituent of cellulose, structural polysaccharide, it is the constituent of, we can say, glycogen, and therefore this glucose is going to be very, very important in our body. That's all about glucose. We'll be discussing other parts of this unit also in coming lectures. Thank you very much for watching.